All right. Once again, I get to announce who we're talking with today. We have two aquatic biologists today. We have Lev Gerstel and Kate Gonzalez. They are aquatic biologists with the State of Hawaii Division of Aquatic Resources. That is a division under the Department of Land and Natural Resources. And they are on the aquatic invasive species team. Now, folks, maybe haven't realized that we have an invasive species response team. We do, and they are fantastic. In fact, Hawaii is a leader in the US for having an aquatic response team for marine issues as well as freshwater issues. So Lev and Kate are on the AIS team. They have been responding to aquatic marine invasive species issues for a while now. They have been active in a new campaign called Don't Let It Loose Hawaii, and that is focused on responsible marine aquarium ownership. And we are going to talk about that intersection today. The talk, gosh, where's the title of the talk is going to be Impacts of Aquarium Releases in Kāneʻohe Bay. And I also wanted to recognize the Hawaii Invasive Species Council staff we have assisting us today, and that is Elizabeth Speed. Thanks so much, Beth.
Thank you for that lovely introduction. So obviously today we're going to be talking about the Hawaiian Islands and Hawaii. And Hawaii is one of the most geographically isolated land masses in the entire world. It's about 2,300 miles to the nearest habitable landmass, California. And this geographical distance and isolation has helped create the bottleneck that allows Hawaii to be home to over 8,000 endemic species that are only found here in the state, nowhere else in the world. And like I said, these species are an absolute powerhouse for the state of Hawaii, a $1.2 billion a year tourism industry, about $13 million in nearshore fisheries are supported by these species. And the reefs around Hawaii, these unique reef ecosystems, help prop up about $350 million in building value by preventing coastal erosion. Now, unfortunately, a lot of these species are under threat. And what Kate and I do is work for the invasive species team. So today we're going to be talking about some of those invasive species. Now, what is an invasive species? An invasive species is a non-native organism that causes harm or is likely to cause harm to the environment, to the economy, or to human health. And you might have seen some of these if you're boating or swimming or going around Kāneʻohe Bay, things like the upside-down jellyfish that Hawaiʻi Fish Pond has been dealing with lately, things like the peacock grouper. These are found all around the Hawaiian Islands. We have numerous invasive algae that we see here in Hawaii, prickly seaweed and gorilla ogo, and things that are found in freshwater, like the invasive catfish and water hyacinth. So these are just some examples of the invasive species that we might find either in Kāneʻohe Bay or around the Hawaiian Islands. Now, how do these invasive species actually get to Hawaii? Well, there are four main pathways that they might take. Ballast water, biofouling, aquaculture, and aquarium releases. The majority of these are going to come here through ballast water and biofouling. About 78% of invasive species make their way to Hawaii through these two pathways. Aquaculture and aquarium releases prop up the rest of them, but today we are specifically going to be talking about aquarium releases. Now, we know that in the state of Hawaii, we have recorded 748 introduced aquatic species. When I say introduced aquatic species, the invasive species cause harm to either, like I said, the economy or to human health or to the environment here in the state. Introduced species are species that don't necessarily belong here in Hawaii, but not all of them cause harm. It's an important difference between the two. So in the state, we have 748 introduced aquatic species. This is almost certainly an undercount. A lot of these are very hard to find. There are most likely more than this. 36 of these have been introduced by aquarium releases, so about 5%, which doesn't seem that high, but we know that 42% of these aquarium releases have happened since the year 2000, and we have records going back to the 1920s. So these numbers are almost certainly increasing as we go on. And the aquarium trade really started back after World War II, when shipping industry started opening up around the world in places like Sri Lanka and in the Philippines and in Hawaii. We know today in the US, about 13 million households have home aquaria that they keep. This is about a $2 billion a year industry that transports around 55 million individual species every single year, which is a lot. That's a lot of chances for aquarium releases back into the environment. Now, DAR has specific strategies to try and deal with some of these invasive species. Really, it relies on 3 pillars. First pillar is prevention. If something actually is released into the water and it gets loose, it becomes an invasive species, it is very, very hard to eradicate. The best step that we can take for preventing invasive species is stopping them from getting here in the first place. If they do manage to get loose into the environment, early detection is critical. The earlier we can find these, the less of an impact they can have, the easier it would be to have a rapid response plan in place and to try and do some management and control, if we can. We remove these invasive species. If not, we do our best to try and manage them in place. So some invasive species that the team is working with here in Hawaii. First are some soft octocorals that we have found in Pearl Harbor that were introduced via an aquarium release, specifically 2 species, Eunomia stolonifera and Capnella spicata. And these have spread to approximately 100 acres of seafloor in Pearl Harbor. And these are highly invasive species that have been recorded smothering reefs in other localities. They're currently covering about 100 acres in Pearl Harbor, but we know that they have wiped out about 60 miles of coastline down in Venezuela, and they have recently been recorded in Cuba and in the Bahamas as well. So it's critically important that we try and contain this to Pearl Harbor and work to eradicate it if possible, which is something that the invasive species team is doing. We are currently planning dive operations for over the summer to try and get a better idea of what these species are looking like and how much area they currently cover at the bottom of the harbor. Another invasive species, or a few invasive species that we are currently working with, are some non-native corals from Kāneʻohe Bay. 3 individual species, Montipora foliosa, Montipora digitata, and Montipora stellata. Now, these were introduced into the bay, or at least were first recorded in the bay around 2018. And community partners alerted us to the presence of these non-native species. And in order to actually go out and deal with them, we needed to justify their removal and show that these corals had invasive properties. Corals are incredibly important to Hawaii, like I said before, to help with the economy, but also culturally. And so we needed to show that these specific species of coral that don't necessarily belong here in Hawaii actually needed to be removed. And there were some questions that came up during those discussions. Isn't more coral on the reef generally a good thing? We're all trying to, save the environment, save the corals, stop global warming. So wouldn't more coral be a good thing? And then also, corals grow very, very slowly. So what are the chances that these corals are actually invasive, or are they just tiny little things in place that we could get rid of, somewhere down the line? So on the topic of isn't more coral a good thing? Yes, but not in this case. We found that these 3 species of coral were actually overgrowing a lot of our native corals, which is not what we want. That is when something becomes invasive, when a species comes in that doesn't necessarily belong here and begins harming organisms that are already here in place. So this showed that these 3 invasive coral, or these 3 non-native corals, were showing invasive properties. They had the chance to become an invasive species. And is invasion actually likely if corals grow very, very slowly? Well, it's not always about how fast the things grow. In this case, we recorded the possibility that these corals were bringing in diseases that our native corals weren't necessarily able to fight off. And so it's not always about the initial introduction that's the problem. It's about the secondary impacts and the ripple effects that the introductions can cause. And so because we answered these 2 questions, we were able to designate these corals as non-native, as invasive, and we were able to justify the removal. And so the removal took place in 2020 and was initially successful. However, unfortunately, we located a secondary outplant in 2024. And this really highlights the difficulty of eradication once a species is introduced. We thought we had gotten rid of the whole thing, and either somebody went and conducted a secondary outplant, or the coral fragmented and rolled somewhere else on the reef and grew into another colony. And so it's very difficult to find these things once they are out in the environment. It shows why continued monitoring should be a priority for all of these projects going forward. And so we conducted another removal in April of 2024. And this one so far, with follow-up monitoring every 3 and then 6 months, has been shown to be successful. So at this point, we think we have removed all of the non-native coral from Kāneʻohe Bay. And with that, I am going to pass it over to Kate, who's going to talk a little bit more about some of the other projects that we're working on.
Just going a little bit more into that timeline of the non-native coral or the invasive coral that Lev was talking about, to highlight how long-term of a problem this has become from an initial aquarium release. Like Lev mentioned, it was first reported by the Cavello family in May of 2020. It was removed later that year. We have continued follow-up surveys ever since that initial removal, until a second report was received in 2023. So that's 3 years later. We've removed additional colonies, as you saw in the past couple slides, and continued follow-up surveys did an initial small removal of a salata, but this is when an additional problem popped up. So one thing Lev did not mention yet is that there was an anemone that was found with these corals in 2020. So it's a Manano anemone known as a notorious aquarium pest. In the initial removal, it was removed using epoxy, marine grade epoxy, which is very common practice for this type of anemone. But it was not seen again until this follow-up survey in 2024. Sorry about that. So the timeline for this Manano anemone specifically, like I said, it was detected in 2020, smothered in what is thought to be removed, and found again in 2024. Since then, we have conducted reef-wide surveys of this reef to see where it actually is. And it is fortunately within a 30 by 50 meter zone, but you can see the anemone on the right here growing within a Montipora capitata colony. So I'm going to go a little bit more into what we have done to respond and why it's so bad. Like I said, currently it's found in one place in Kāneʻohe Bay. You can see why this anemone is tied to this non-native coral and why it was initially found together. On the map on the right, all of the yellow circles are the original coral removal sites. And then all of the purple stars are the survey points for the Manano anemone from 2024. So you can see how it's surrounding this non-native coral site and then also moving down with these further non-native coral removals. It's also found in Pearl Harbor near Fort Island, but we think that this was likely due to a separate aquarium release, really highlighting the problems that can come from aquarium releases. And in Pearl Harbor, they haven't done additional surveys as of yet, but it is estimated to have doubled within just 1 year in distribution. To really highlight on Reef 8 where we have seen it and what we have done, on the left is the tracks of a survey we completed to really scope out where the anemone is. We covered the entire reef with a fine-tooth comb, making sure that it wasn't anywhere. We also did preliminary surveys of surrounding reefs to make sure it had not spread. And on the right, you can see the small area that it is localized to currently. But keep in mind that this spread has happened from 3 known polyps in 2020, and now it's 2025, so about 5 years since that initial removal, it has spread to this zone. And it's that red polygon within this big zone that we survey and we take photos of. A bit of a background on why this anemone is such a problem. It's able to sexually and asexually reproduce. It's highly fecund and it reproduces very rapidly, as you can see as how much it has spread in the past few years. And it doesn't really have a huge preference for habitats. So we found it on a variety of hard substrates, rubble, dead and live coral, which is the problem. In Pearl Harbor, it's seen growing on mangroves. I'll go into more why we know it grows on glass or is okay on glass from some small scale trials that we completed here at Anuenue. It also settles into these really hard to reach crevices on the reef, which is why we think that it took so long to detect it again after the initial 2020 removal. You can see on the right, it's really in these pukas on the reef. So if you were to swim over the reef, not knowing what you were looking for, you would not necessarily see it. It's in these dark places. Oftentimes you need a flashlight to really see it or to know exactly what you're looking for. Going into more of why it's such a concern. When it gets stressed out, it may spawn. It also grows very rapidly, like I've said, and competes for and smothers native coral. So it competes for space with the native coral, and also we have seen it overgrowing and smothering it on this reef. It is not known to sting humans, fortunately, but it does sting other species. And it is able to freely unattach from substrates and move. So we have seen this firsthand in small tanks when we've done lethality trials. But this is why we are also very concerned about it on the reef, that if we're agitating in any way, if someone were to perhaps kick it up, it might just move away to a new spot, or it might, like I split in half, leave a disc behind, and then become two full anemones and really spread that way. On the right, you can see it's the small polyp of the anemone. So we see it as these small polyps, and we also see it just really carpeting these holes in the reef. How did this anemone spread? I mentioned that it was an aquarium release with this non-native coral, but the important part is that it was likely unintentional. So even if this non-native coral was released intentionally, it could have been intentional or unintentional. The Manano anemone was likely an intentional hitchhiker to this release. So it's often found in aquariums, and it tends to like hide within these aquarium species. So what could have happened is that the person who released the non-native coral either knew that it was there and wanted to get rid of the aquarium because it was causing problems, since the anemone is such an aquarium pest, or it was unintentionally hiding in the non-native coral and they had no idea. It also does biofoul. So in Manila Bay, it's been discovered in several biofouling communities, meaning it could spread via hulls of different ships, kayaks, anything that's touching it. And like I said, it also freely moves, so it can just pick up and move to a new location. Why is this aquarium invasion so bad? It really is a nuisance species, often compared to Aiptasia, but considered worse. The reason it's worse is that aquarists who find this pest within their system often have to get rid of the entire system and start anew. There are ways to get rid of it in small scales, including what's called a Manano wand, where you zap the anemone. But aquarists have reported that you have to do this repeatedly to get rid of just a small anemone. So when it really starts to overtake a reef, often the entire or overtake a tank, sorry, often the entire tank must be gotten rid of and started from scratch. So it multiplies so quickly that this often has to be the case. In greater literature or from aquarists firsthand, they have said that they have seen it in 3 weeks go from 4 to 50 anemones within their tanks. And like I said, we've tried marine grade epoxy and other smothering treatments, and because they can just pick up and move, they will just get up from under the glue or the epoxy and go around it, and they'll live. So this is the trials that I was talking about. We did small scale lethality trials on Sand Island because we are trying to figure out the best methodology for treating this anemone on the reef. You can see on the left, the anemone migrating around the tank, including in the control where we did not treat it, and in the epoxy tank. So it was able to escape the epoxy and move around the tank. And then on the right, it actually grew either through, we didn't have a camera set up, but either through the epoxy somehow, or it picked up and moved and just settled right on top of it before it had hardened. So it did not die from this epoxy treatment. And for context, epoxy is often used by our team in settings to smother an invasive species that is not mobile. So we've used it for corallimorph species. We use it to permanently attach things to the reef. It's not supposed to be a lightweight method. It's supposed to be pretty hardy, and it does not work for this anemone. What we did try that was successful was chlorine with tarping. So this will likely be our chosen method. But we were able to cover the anemone so it's not able to move, and then treat it with chlorine. So after 24 hours, we did see that it did die. So moving forward, this will likely be our plan to this very small but mighty aquarium release. You can see on the right how small the polyps can be sometimes. We did collect some to do heat trials, or Terry Work did heat trials with of the anemone. It was even hardy then, so heat was proven effective for other prominent invasives, and it did work on the anemone, but at higher temperatures and for longer periods of time. So it really is just a difficult species to get rid of, and highlights why aquarium releases can be such a problem, even when the species that is released is not the intended release.
Okay, so we've told you a little bit about what we do, but it's also important for us to tell you what you can do, because this really is a community effort. I talked before about how the non-native corals were first reported by some community partners. There's only so many of us, and there are a lot more people out there who are on the bay every single day, fishing, swimming, living their lives, and it's important that everyone knows what they can do to help us control invasive species in Hawaii. So first thing, if you see something, report it. That's how we get reports about these invasive species and how we can put together these management plans to try and control them. You can turn any illegal pets into amnesty stations, and you can also turn in unwanted pets into amnesty stations. A lot of these aquarium releases happen because people decide they don't want their home aquariums anymore, and they say, I'm just going to toss it into the water. Don't do that. That's how we get invasive species. You should instead turn these into amnesty stations that are located all around the islands. And you can help spread the word about the Don't Let It Loose program. This really encourages people to not release their home aquariums into the water because of this specific problem, because those home aquariums turn into invasive species. There are drop-off stations all over the island at local humane societies, at municipal zoos, or at any of the Hawaii Department of Agriculture's plant quarantine offices. And we know that people do use these resources. An illegal coconut crab was turned in Salt Lake in 2014 to a drop-off station. Somebody surrendered an emu in Hilo. I'm not really sure how they got this emu out here in the first place, but they surrendered an emu back in, I think, 2015 or 2016. And then a snake was turned into the Honolulu Zoo under the amnesty program in January of 2023. So people do use these reporting and these surrender stations to turn in illegal pets, or home pets or aquariums that they don't want anymore. So we encourage everyone to talk about the Don't Let It Loose program and to utilize these resources to help us control invasive species in Hawaii. And if you have anything that you would like to report, you can contact us. Our email is over there on the left slide, along with our phone number. Or you can go to don'tletitlooseh.org, where we have a whole bunch more information about reporting invasive species, about turning in invasive species, and about how you can help us control invasive species in Hawaii. You can also scan this QR code and it will take you to that website. And with that, I believe we are finished, and we will turn it over to you with any questions that you might have.
Thank you so much. I'm gonna give you props right now. Right, we can leave the screen up for just a moment, and then we can take a look at each other and see if there are any questions. Yeah, there's one posted here. Are there any statewide or coordinated monitoring efforts for detecting invasive anemones across the Hawaiian Islands? I'll stop there.
Yeah, so this is what Lev mentioned earlier. We really rely on the community and people who are out on the water all of the time for these reports. We do have, like Chrissy said, a good team, and we work really hard, but there are only so many of us, and only so much ground we can cover of the water. So we know the sites that we monitor, we know where, but this, like the Reef 8, that site was from a report, so we would not have known about it otherwise. So we really rely on the public and on people who are out. So that's why we encourage everyone to contact us with any reports. We will reply to emails. We even also check, we have an iNaturalist page that we check, but the best way is really just to contact us if you have any sightings of anything that you think is even non-native besides the anemone.
Yeah, great. If you wouldn't mind, we can take down the screen. We can see each other, and then I'll ask sort of the follow-up question to that. Thanks. are there known habitat types, harbors, tide pools, offshore reefs, where these species are more likely to establish and go undetected? Either you want to take that one.
So when it comes to aquarium species, that also kind of feeds into why people release these aquarium species. Sometimes it's just because they don't want their home aquarium, and they might go and dump it in what they think is a good environment for it. We also know that some people have some more, I guess, nefarious ideas about aquarium releases. We know that there was somebody back in the early 2000s who released a whole bunch of clownfish into Kāneʻohe Bay because he wanted to ranch them and sell them for profit. And he actually did some research and put those into what he thought was a good environment for them. So it depends whether it's an accidental release, somebody is transporting some fish and it just happens to end up in some kind of environment, whether someone puts in the research to put this species in what they think is a good place for it, or whether it's just a release of opportunity. It's what's near their house and they say, I just want to get rid of this and I need to put it into the ocean, and that's where it ends up. So it really varies from release to release, whether or not it ends up in one specific environment.
Yeah, thanks. Kate, if you want to add something to that, welcome to. Now I'll move on
I
to
think
the
that
next
was
question.
great answer. The only thing I would add is that that's a great answer for aquarium releases. If you're talking about in general where species are more likely to establish and go undetected, it is likely harbors, because that's where a lot of them are introduced via ballast water and biofouling, which is one of the biggest vectors of invasive species. So we do see a lot of early detections in harbors. It's also because people aren't necessarily in these waters all of the time. Probably a little less likely on like offshore reefs, but again, like Lev said, it really depends on the introduction.
Yeah, definitely agree. I'll just chime in here on your Pearl Harbor introduction. That 80-acre footprint of the Eunomia and the Capnella there. I think probably a water body of opportunity. Certainly, it wasn't a pristine water body. They were just looking to try release, but that was discovered because they were doing a survey. Navy had to do a biological survey. So again, thank heavens they found it. We'll see whether or not it's in time for their eradication efforts. And you folks, your work trying to help survey outside the harbor, if you want to speak to that for a moment.
Yeah, so the Navy has really been taking the lead on the eradication after they detected it in Pearl Harbor. But our team, hopefully this summer, it's really moving along. Finally, we got access to the zone outside of the known infestation of Eunomia. So we will be conducting delimiting surveys of the area outside of this known zone to make sure that it has not spread any further. Really, the concern is that it keeps spreading and then enters state waters and it keeps going from there. So we'll be looking at that area outside in the coming months.
Yeah, thanks. Anybody have questions? If so, put them in the chat. And I will continue on with some of Beth's questions. What detection tools or methods, such as maybe eDNA or diver surveys, are most effective for early identification of invasive anemones, for example, in Hawaii? You're on mute, Kate. There you go.
Yeah, sorry, we were just making sure who's the answer. I'd say for early detection specifically, eDNA is likely the most effective. There's been some great work recently from labs at UH who are working on detecting species via eDNA, but it also depends on, if the markers are already available. So it's difficult when the genetics are a little iffier when there's not a marker yet. The only reason I say that though is because diver surveys, I think, are difficult to cover large spans or large areas of water in. So just for that early detection, it's really, yeah, I guess for early detection, it's like the widest area you can cover. So that's also community reports, and then eDNA also could do like aerial surveys. It really depends on the detection, but I'd say most of the time they're coming from reports.
Nothing to add, Lev? Okay, yeah. Yeah, you know, I was at a webinar, actually it was a talk in person, it was an in-person talk, where a researcher at UH was using eDNA to detect chondria, that's the invasive seaweed that's spreading in the northwestern Hawaiian Islands. And I was amazed that they were able to get water samples from the surface, and 30 feet down is where the chondria was. So it remains to be seen how much anemone you need to be able to detect it, and therefore how much water you might need to sample. So yeah, fun times ahead. Let's see if there's other questions. Any other questions in the chat? I have one, if you don't mind. You know, I was at an event that DAR had some imagery set up where it was a video of the reef, like a transect of a reef, and you could go back and review it. Is that a possibility for maybe underwater drone work, or how does that work for you folks? Are you able to do that? Oh, or should we buy you a drone and you can find out?
Yeah.
So we Kate showed, I believe there was an image in there. We're doing some photogrammetry, which is really highly detailed photography of the reef, and we're able to build 3D models of the reef once we do this. It's really useful for seeing the complexity of these reefs, because a reef is not a 2D structure, but a photo is 2D. And if we can build a 3D model of this and really see all of the biological processes and I guess the building blocks of what goes into making a reef, we can understand why some of these invasive organisms might inhabit specific parts of it. And then we are able to review those photos and see that reef from more than just a top-down perspective. And we don't necessarily need to dig a camera into every single little puka to see everything. We can do big flyovers of large spaces of reef, and then use all of those individual photos that we take to turn that into one large photo mosaic that we can then go back and review to better understand what is going on that reef. We use that as well for a lot of our restoration practices to see changes over time. So that's been a really useful tool that we have started to ramp up in recent years, our use of it. It's been very helpful in all aspects of our work.
Right on. Thank you so much. Well, last chance for anybody to ask questions. All right. Thank you so much for bringing your story and all of your expertise to this work in Hawaii, Lev and Kate. Really appreciate what you folks do. I look forward to watching and hearing about the eradication attempt as it moves forward, and go Reef Warriors, go save our reef. So